Okay, hi. So we're going to go through D3.1 reproduction first. It's pretty chock-a-block, like there's a lot in it. So when I made my page, I had to go pretty small. So my general guidance would be small. I'm going to make really tiny diagrams at the top. So if you want to fit it all in one page, especially if you're HL, I would say go tiny. If you're not concerned about fitting it on one page, happy to use front and back, then you don't have to stress as much. Okay, so let's start with what we want to clarify is that when we say reproduction, this specific area is very much looking at sexual reproduction. So let's just quickly make sure we're clear on the definition of sexual reproduction and on its benefits. So what we want to be careful of, and this matters when we get to the plant material, plant stuff, is that we say that sexual reproduction is the merging of two unique gametes. Does not have to be two unique parents. So technically, if you have a hermaphroditic plant and both gametes come from the same plant, but they are two different gametes, it is still sexual reproduction. So it is the fusion of two genetically unique gametes. So that's kind of how we want to define it. And then the rest is pretty straightforward. We know this from our evolution standpoint, but the benefit of that is that it creates unique offspring. So the offspring are unique both to each other and to the parent. And that increases genetic variation in the population. And then from there, you should be able to access all of your A4, D4 knowledge and link that to natural selection. So increased genetic variation, more likely to have a beneficial mutation for environment, and that's going to increase that. So that's our definition of sexual reproduction. So that's a good kind of starting point, is that we know what that is. So there's 3 reproductive systems that you have to know. So I'm putting them kind of next to each other, even though they're humorous that the flower is next to the penis, but that's how we're going to roll with it. So we're going to do a little diagram of female, a little diagram of male, and then a little diagram of a flower. Again, because of how much is in this topic, I would advise these diagrams be on the small side if you can manage that. And you can basically divide your space. So I might draw them not super tiny on my board, just so it's easier for you to copy. But just note that if you want to fit it in, make yours on the small side. Okay, so let's look at the 3 reproductive systems you could have to draw. So all 3 of these are draw commands. So draw, label, and function. So we're going to do female reproductive system, we're going to do male reproductive system, and then we're going to do flowers. And I wanted to say, instead of just say plant reproductive system, this is specifically how angiosperms, which is flowering plants, reproduce. So not all plants reproduce this way. So we're going to say angiosperm reproductive system. I'm just going to, hopefully you did a better job dividing into 3s. I'm going to give a little bit more space for my angiosperms. Okay, so now let's go through the main parts. So I'm doing these as fairly simple diagrams, just because the key is to really know the most important things, and the simpler we make it, the easier it is to remember. So for our female reproductive system, we're going to draw a uterus that kind of closes in with the cervix. Do some lining there, a vagina. So we've got uterus, we've got endometrium, we've got vagina, and we've got cervix. So those are all essentially kind of linked things. And so for each thing, we'll go back and add like 1-line functions. So just trying to keep it pretty straightforward. The other big part, of course, is going to be the fallopian tubes and the ovaries. So there's my fallopian tubes, and here at the end of those is the ovaries. So that's essentially all you need with the female reproductive system. A little bit simpler, which is nice. because we have less glands involved. And then ovary. So I'm going to just put essentially a one-word description of what as quickly as possible in terms of what's happening at each one. So ovary, we're going to put in, say, ovulation. So that is going to include making and maturing the egg as well, and then releasing it. Fallopian tubes is going to be fertilization. Vagina is going to be intercourse, so that's going to be the delivery of the male gametes. Cervix, kind of hard to manage that. We're just going to put gatekeeping. So that's my one word. So essentially trying to make sure the baby stays in during pregnancy, but the sperm is allowed through. So managing what goes between these two areas. Endometrium, we're going to put implantation. So that's the lining, and uterus will put pregnancy. So again, in one word, that's the function. If you were writing it on an exam, obviously it probably would be beneficial to use more than one word. So I'm trying to describe it in the shortest way possible, but it's always beneficial to be a little bit more detailed. Okay, so let's move into the male reproductive system. So I'm going to start with the ureter, like so. On top of the ureter, we're going to put the bladder. The bladder is part of the excretory system, but it is worth kind of noting it here just to kind of give us some context. We're going to put a couple of our glands. So we're going to put our prostate gland below the bladder. And then next to that, we are going to put some seminal vesicles. Okay, then we need to actually make and develop our sperm. So we're going to add some testes down here. The testes need to then be connected to the actual glands. So that's going to happen with the vas deferens, like so. So we draw the male reproductive system like a beautiful butterfly. And then we need to add the kind of outer areas. So we need to add the actual scrotum, which is going to be around the testes, and then the penis, which is going to be around the ureter. So those are the external structures involved. Okay, so let's go through and label these. So the ureter here, then we're going to have the testes. We're going to have the scrotum, the penis. We're going to have the vas deferens. And then we're going to have our two glands. So we're going to have our seminal vesicles. And then we're going to have our actual prostate. So if we go through and do the same thing we did with the female, which is can we say in essentially one word what they do? Oh, I forgot the epididymis. Too many parts. There we go. Okay, so you see more colors, more parts. So male reproductive system has a little bit more complexity in terms of number of different locations. So if we have testes, that's going to make the sperm. If you're HL, you'll need to know the word spermatogenesis, which just means sperm making. If you're SL, you don't need to know that term, but hopefully that makes you can just write making sperm. So that's going to happen here in the testes. The epididymis here is just for storing of them. So epididymis is going to be storage. It may not have a Y there. It's fine. Okay, so there's going to be our storage. I'm going to correct it because I don't want to spell it wrong. That's better. Okay, there's our epididymis is for storage. Penis is also going to be for intercourse, for ejaculation of the semen, so either of those. The scrotum is going to be for cool temperature, so I'm going to put thermoregulation. So it's going to keep the temperature lower by having those outside the body. Ureter is going to be for delivery. Vas deferens is going to be for transport. And the prostate and the seminal vesicles together, put them in semen. They do have a slight difference. So seminal vesicles are primarily going to make like the carbohydrates and proteins and the nutrition that's needed for them while they're in the semen, whereas the prostate is going to be in charge of maintaining the pH. So it's going to actually make sure that semen is slightly alkali, which is helpful for them to swim. So I'm just going to put pH and I'm going to put semen. Okay, so those are, again, one-word definitions. If you want to go into more detail, go back through your original notes. We're just trying to, again, make this a consolidating, so making it a little bit shorter. Okay, so we're going to go from there now into the angiosperms. So for our angiosperms, these, of course, are hermaphroditic, so they are going to have most of them, not all, but we are going to have both male and female parts on the same one. So there we have my stigma, my style, and my ovary. Inside the ovary, we're going to have ovules, and we'll show multiple ovules. So all of those together make up the female part of a flowering plant. So everything here is the carpel or the pistil. So carpel or pistil is all of those parts. Okay, so now let's add some male reproductive parts as well. So then coming off of here, I don't have a lot of space. When I drew this in class, I drew like the world's most giant. Oh, I probably should have done my labels after I finished my drawing. Oh, well, you live and you learn. There we go. So there's some filaments. Then we're going to do some anthers on top of those. Anther and filament. And together those make up the stamen. Okay, so the stamen is anther plus filament. Then we need some beautiful petals. Ooh, petals. It's not the prettiest flower due to some of my poor decision making about when to label. And then we're going to draw some sepals. So sepals here, and then we'll label our petals. Okay, so now we're going to add our one word of what everything does. So same kind of idea, single, just a memory prompt of what they happens. So stigma is going to be our sticky. So it's going to put collection. The style is going to have the pollen tubes. So that's going to be transport. I'm going to add a little note there on the transport thing. So remember, these only form if the self-incompatibility alleles are different. So pollen tubes are going to be sensitive and check and try to eliminate forming if it's coming from the same plant. So pollen tubes for transport, but they only form if there is self-incompatibility is checked and checks out okay. So that's what they're going to be transported in. The ovules are the gamete, so that's going to be the female gamete. And then the ovary is going to house that and then ultimately become the fruit. So it's going to have the ovules inside of it, and then once they're fertilized, it's going to ripen into fruit. Okay, so that's all of our female parts. Our anther then is going to also be the other gamete, so that's going to be the male gamete. And the filament is basically just going to be for position. So the filament is going to ensure that the anthers are up high. So gamete position. So if all of these are too short, go back and learn longer explanations. The idea of this is just to see it and have a word to prompt your memory. So petals are going to be in charge of attraction. and sepals are going to be in charge of protection. Okay, so that's every part of all three reproductive systems, labeled, drawn, and with a single word prompt to try to help you remember what they are actually doing. So I'm going to actually kind of continue with the thirds. So the next section is going to be in thirds as well, because we're going to then go, okay, while we're on female reproductive system, how are eggs made? When we're on male reproductive system, how are sperm made? And then while we're on plants, we'll talk a little bit more about how they actually develop seeds. This little section here is HL only. So I'm gonna do a little HL minute. This section's a little bit harder, it goes back and forth. The next section's like all SL, all HL, whereas this jumbles a little bit more. So SL, you are going to need to know this here, so you can fast forward a little bit and join us when we get to seeds. HL, we're going to take a second and now look at the actual making of the gametes and oogenesis and spermatogenesis. So we're going to pick that up. So I'm going to erase, and you're just going to keep going below on your page. All right. So like I said, I'm going to kind of keep doing the same thing. I am going to label here that now we're going to look at making gametes in both the male and female reproductive system. And I want to make a nice clear label for you guys that this is HL only. If you're HL, it doesn't necessarily matter that you write that because you have to know it all. And if you're SL, tune out. So it's really just for me to remember to tell you that. Okay, and then we're going to leave that space over there for seeds. Okay, so then when we look at making gametes, the processes are similar but somewhat unique. So we're going to kind of divide our space and look at spermatogenesis versus oogenesis. So oogenesis versus spermatogenesis. And just to make sure because one thing you could be asked is to compare them. So just in case that was asked, let's just quickly refresh ourselves on what are some of the kind of key differences. So with oogenesis, we're talking about making one large egg. And we're doing that monthly. When you say large, hopefully that will prompt you to remember that it's large because it has the food stores in it. So we have the uneven cytoplasm division so that we get this one large egg. Spermatogenesis is going to be essentially millions of tiny sperm constantly. Daily, hourly, depends. So it's different factors, but essentially it's just kind of happening all the time. So this is kind of constant, this is monthly. So again, number, size, all of those things that are different. Okay, so then let's go through the actual process. So I'm actually going to start with the male one because it has more steps, but it's a little bit more straightforward in terms of there's not kind of the gaps between it. So we're going to start with a spermatogonium. Okay, spermatogonium, and that is going to be a diploid germ cell. So that is a diploid germ cell. That diploid germ cell is then going to undergo mitosis. One of it is going to replace, one is going to keep going. So it's going to now undergo mitosis. One cell continues, one cell replaces that one. So there's kind of a constant number of that. After mitosis, it is now a primary spermatocyte. It is still diploid at this point because mitosis does not divide chromosomes. Then it is going to go through meiosis 1. It is now a secondary spermatocyte. And it is now in So it is now a haploid cell, but it still has the sister chromatid. So if you can kind of connect this back to what you know about meiosis. So we've done one round of meiosis, still has the sister chromatids. Next, we're going to divide again in meiosis 2. And after meiosis 2, it is now a spermatid. They have too many names. So a spermatid would basically mean the nucleus is done, but it's just a cell. So it needs to then undergo differentiation or specialization. So cell specialization, which means it needs to get its actual little tail and get all of its structure. And this is done with help from Sertoli cells, which are sometimes called like nurse cells. So the Sertoli cells are basically going to provide the nutrients and help with that process. So they're kind of these larger cells in the seminiferous tubules that are in charge of doing this. And this process happens if you have space in the seminiferous tubules. Just in case you were to see that term, you can just remind yourself that that's where this is taking place. So after this, they are now reaching their final destination. So they are now sperm. You will sometimes see spermatozoa. So just in case you see that, you're fine to call them sperm, but in case you see the other term. So that is kind of the end of the process. So that one is continuous, which makes it a little bit easier to outline it kind of step by step. So you need to know at every stage is a haploid or diploid and what's going on to make that happen. The female oogenesis is a little simpler, a little worse. So simpler because it doesn't actually go through as many steps all the time, but a little bit more complicated because it has some pauses. So starts with a similar thing, which is an oogonium. That is a diploid germ cell. That diploid germ cell is then, and we're gonna write when it happens, also gonna go through mitosis and replace. And it's going to start meiosis 1. So it's actually going to enter and stop in prophase 1. It doesn't really make much progress other than the pairing of chromosomes. So it is essentially beginning that process. So it is now a primary oocyte. Still 2N because we haven't made much progress. This primary oocyte is in prophase 1. So I was trying to write tiny, so I wrote in P1 for prophase 1. If you're not going to remember that, just write bigger, find space. But that's just to remind us. This is going to happen in utero. So this process happens before the female infant is even born. So this is happening right away. Next, that primary oocyte is going to finish meiosis 1, and it's actually going to start meiosis 2. So the male's one is nice because it's a little bit more clear cut. The steps stop in between and everything has its nice new name. So this is now going to be a secondary oocyte. And it is now in, so it is now haploid, and it is stopping in metaphase 2. So it makes another step forward. So it is going to pause there. This process is what happens after puberty, and it happens monthly. So this is essentially the menstrual cycle, which we'll do next. So this process is what's happening during essentially the follicle development and so forth, is the developing of that. Okay, then it's going to stay like that, and it's actually only going to finish meiosis 2 if it is fertilized. So that is going to only happen at fertilization. So if the egg is released and sperm joins it, it will complete its meiotic journey, and at that point it will become essentially a mature ovum or egg. But is essentially already a zygote. So a little bit confusing because by that point it is actually a zygote because it's already been fertilized, but its own nucleus will finish the process before it joins with the sperm nucleus. So it does have a little moment in the sun to be a mature one. So again, slightly more confusing with the females because of the need for the actual kind of stopping. So that makes it a little bit more complicated. Okay, so let's go from that. SLs, welcome back. Okay, so now we're gonna do making seeds over here. So this is kind of the last piece of our plant area, is making sure we knew that. So the seed is gonna happen as the result of cross-pollination. So the goal is always cross-pollination. Ideally. So they can self-pollinate, and there are instances where plants will self-pollinate, but self-pollination is not going to provide as much genetic diversity. So cross-pollination is an increase in diversity, and there's mechanisms in place to help that happen, so they can adjust timings. They use self-incompatibility measures, and some plants use single-sex flowers. So those are just, if you were to be asked about some mechanisms, it would be like developing the male and female parts at different times. It would be using flowers that are all male, all female, or plants. And then the last part would be the self-incompatibility mechanisms. Okay, so then assuming cross-pollination happens, the pollen tube will form. So if it meets its requirements, the pollen tube forms. And then the actual pollen double fertilizes the ovule. It mentioned this in the textbook, but it's not really mentioned in the syllabus outline in terms of this concept of double fertilization. But essentially the sperm has 2 nuclei, each ovule has 3 nuclei, 1 and 1 just become the actual like embryo, and then the other 3 join to make a triploid endosperm food source. So I'm not gonna put all that up because it doesn't look to me like that's in the scope of what you need to know. But I just wrote the word double fertilization just to like prompt your memory that it's a little bit more complicated than that. Okay, so then you need to know what the actual seed looks like. So there is my actual seed. There's the seed coat. You might see the word testa for that, but it's no longer written in the syllabus, so I'm just gonna call it the seed coat. And then here's the little plant embryo, and particularly when it starts to grow, you're gonna see this part here, which is gonna be the radicle. And radicle equals root. So it is going to be the part that is going to go downwards. And then you're going to see another part that is going to be what is going to ultimately become the stem, which is the epicotyl. And then all of this inside here, a little bit confusing because it would be called prior to germination, it would be called the endosperm, which would just be the food source. And then after germination, it gets called the cotyledon. And that's once it's essentially activated and the plant is growing and developing, that now takes on a slightly different role where it's going to actually kind of continue to have to generate food sources and growing it. So essentially endosperm becomes cotyledon. They are the same part of the seed. We just call it at different times. So before actual germination, we would just call all of this the embryo. But once it starts growing, we can differentiate the radicle, the epicotyl, and the cotyledon. Okay, so that is the parts of it. Then we are going to have what needs to happen to this beautiful seed. So the first thing that needs to happen before germination is it needs to be dispersed. And that means it needs to go away from the parent. And the reason is a couple of different things. First of all, the parent is already using the environmental resources in that area. So by moving it away from the parent, it has more likely access to resources. The other thing is, again, genetic diversity. If we move it away from the parent, it decreases the likelihood of kind of plant inbreeding, of it then mating with its own parent. So good to have dispersal. Then from dispersal, we then have germination. And that is when it's going to start to develop into the parts. For germination to happen, the most important thing is for it to get enough water. So basically the whole process is triggered by water. So water is going to activate some enzymes that are going to break down the endosperm so that they can use the starch stores for cellular reproduction, and that facilitates growth. So the water needs to go in, which is imbibition, which is such a fun term. But remember, if you draw a blank, just say water enters. If you can often get points without knowing all the terminology. The other thing it's going to have is access to like appropriate pH in the soil, sunlight, things like that for it to be successful at growing. So the conditions have to be right. So it will only do this under kind of ideal conditions, which include things like having the right temperature for the enzymes that are needed to drive the process. So temperature, pH, things like that are going to be necessary for germination to be successful. Okay, so that's all things for our plants. The next part is going to be SL as well. So we're going to do another SL little section here, and then we're just going to leave at the bottom for the remaining HL stuff. So SLs, hang in there. One more section for you. All right. Okay, so we are going to do the menstrual cycle. So I'm going to do it because I'm trying to do it a different way than we did in class. So in class we did the giant kind of hormone curves. It would still be good to go back over that just so that you could interpret it if that were given to you. But this time I'm going to do it as a chart because I find the curves a little bit confusing for some students. So I want to be very clear of like, this is the hormone, this is who releases it, this is when it's released, this is what it does. So a real kind of hormone focus. And then over here we'll just draw what's going on in the ovaries and in the uterus during the stages. So that's just a different way of doing it, again, to try to mix it up. So we're going to look at female reproductive hormones. And actually 2 of them on my chart are HL only. It doesn't hurt you to learn them. It's good to know your hormones. So SLs, if you dramatically want to, you can leave them off, or you can just write them down and put a little star to remind you. You don't have to actually know those. So for our chart, we're going to say, what is the name of the hormone? The next thing I want you to know is who releases the hormone. The next thing you need to know is when is that hormone released? And then the last thing you need to know is what are the actions of that hormone. And then let's leave a little bit of space for our menstrual cycle over here. So I'm just going to like remind us not to use all our space. So we're going to use a little space over there to the side to do a little menstrual cycle. So monitor your spacing so you still have room for that. want a bit more space for my win. Okay, so the first one I'm going to start with is one that is not actually needed at an SL level, so you can go straight into the ones that do the menstrual cycle. But for HLs, you do need to know what starts this whole thing in action, which is the GnRH, so gonadotropin releasing hormone. So GnRH. With all of these, it's hard to say. I would try to know their full name of like follicle stimulating, luteinizing hormone, and so forth. If you draw a blank and you use their initials, it's likely you'll just not get what would have been a possible point, but you could get full marks without it, so I wouldn't like super stress about it. But if possible, I would know what they stand for, would be my recommendation. Okay, so that is actually going to be released by the hypothalamus. And that is going to be released at the beginning of puberty. So that is essentially what's going to be responsible for puberty. We talked in class that it's responsive to a protein called kisspeptin, which is essentially released as we grow. So it's kind of related to growth, nutrition, all of these factors. You don't need to worry too much about that in terms of that term isn't in there, but you do need to know that it's related to puberty. And so what are its actions? So it itself does not lead to puberty, but it is going to then trigger the release of both FSH and LH. And we will do more on those if those are going to be our next step. So that triggers the release of the next things. Okay, so that's our first hormone. First sex hormone. And that actually does in males as well. So I'm doing female sex hormones because that links in with menstrual cycle. But this first part, that part is the same in males as well. Okay, so now let's keep going with that theme. So if that releases FSH and LH, let's do FSH next. So next hormone is follicle stimulating hormone. And that's going to be FSH. And again, if you remember it, great. If not, call it FSH. Okay, who releases that? So that is going to be released by the anterior pituitary. You don't really have to distinguish anterior versus posterior, so I'm just writing A and P, but if you just remember pituitary, you should be fine. And in fact, if you don't remember, better to just say pituitary than say the wrong one. So it's going to start at puberty, and then it's going to be released every month as part of the menstrual cycle. So the first kind of thing it does is at the beginning of puberty, and then it's going to be monthly at about days 3 through 14, sorry, not 13, 3 through 14 of the cycle. So that's going to be in our follicular phase. So like I said, we're going to do the hormones first, then we're going to come back and draw the actual stages and clarify that. So that's in the follicular phase after the kind of menstruation period is ending. So it'll start rising after about day 3 all the way through to ovulation, and then it will be suppressed by progesterone. What does it actually do? It's going to stimulate follicle development or egg maturity. So it is going to tell your little primary oocytes. One of them is going to be the chosen one. It's going to tell the follicle cells around that to grow. It's going to tell the actual oocyte to finish its meiotic journey and become big and developed. So it's going to be kind of working with that individual one. Okay, so that is going to be what FSH is going to do. We'll talk about it later, but it does also play a role in IVF, which I think is like in my HL section of SLs, you do have to know it. So go back over IVF as well. Okay, so then that is going to be follicle stimulating. It also is going to trigger the release of luteinizing hormone. This is probably the harder one to remember what that word is. So LH. So LH, we're going to use the term luteal phase when we do the actual cycle. So LH is going to start the luteal phase because it's going to cause ovulation. Okay, so that is also going to be released by the anterior pituitary. It is also going to be released from the start of puberty, and it's going to play a role in kind of that part as well. But its primary thing is that it's going to surge between days about 12 and 14. So it'll have a little bit of increase leading up to that, but really goes a huge surge. So all the others are a little bit more gradual. This is the one that's going to go. So massive surge days about 12 through 14. And what is its purpose? Ovulation. So it is going to cause the release of the egg. Okay, so those are my initial ones. Once this follicle is developing, that is going to trigger the release of the next thing. So as the follicles start to develop, it is then going to trigger the release of estrogen. So they do use the term estradiol, which is a specific type of estrogen. So you can, I'm sure you would get points if you say estrogen. That's the broader category, but some estrogens are involved more in secondary sex characteristics, and some are specifically involved in the menstrual cycle. So this is the one that is specifically involved in the menstrual cycle, and that is part of a broader category of estrogens. So just to clarify that, in case particularly if you see the term estradiol, that you recognize it, but I think you're fine to use estrogen if that makes more sense. So this now is going to be released by the ovaries, and in particular by the follicles. So it's released by the growing follicles, at least initially, and then it can be released by the ovary itself, kind of in the second phase of the cycle. So that is going to be released then. When does it done? So first of all, it's going to be a big one in kind of puberty. So it's going to play a really big role in kind of the secondary sex characteristics, particularly these ones, the more general estrogens. And then in terms of the cycle, it's actually going to surge. What was the best way I said it? It's kind of rises at different times. So it's going to be kind of rising in the follicular stage. And it will actually go up again in the luteal phase. So it's really our kind of primary sex hormone that does quite a lot of things. So it's a little bit harder to track. It's going to play an important role in this follicular phase. That's probably the most important one that you need to know, because it's going to trigger the release of more FSH and LH. But it doesn't go all the way down like the other two do. So the other two go right down in the luteal phase. This is going to continue developing in the luteal phase as well. So it's kind of all throughout. Okay, so what does it do? It does kind of three things that I want you to know. So the first one is it's going to be responsible for secondary sexual characteristics. And as a result, it has a broad range of effects. So it can be important for like bone development, all kinds of things. So it's kind of overseeing that whole post-puberty female development. In terms of in the cycle itself, it is going to positively reinforce FSH and LH. So it's going to positively feed back. So as the follicles start growing, the estrogen release triggers more of these. So it is going to increase the levels of FSH and LH kind of later in the follicular phase. So once the follicles are big enough and estrogen rises, it will trigger that. And it also is going to do some thickening of the lining. It'll get even thicker once progesterone gets on board, but it will begin that process of thickening the lining in preparation for pregnancy. Okay, so that's estrogen. That's probably our most significant and diverse one. Most of the others you can kind of track to more of a single impact that they have. And then the two more, last one for SLs is progesterone. So progesterone is going to be released from the ovary, but specifically from the corpus luteum. So it's the corpus luteum in the ovary. So just like we say ovary here, but we want to clarify it's actually the follicles releasing the estrogen. And I think the corpus luteum will also release some estrogen as well once the follicle has released. But really the focus on the estrogen is on that first part of the cycle. Progesterone is going to be the corpus luteum in the ovary, and then during pregnancy, the placenta will take over. So it'll end up releasing it through the placenta. When is it released? Basically following ovulation. So it's going to be days kind of 14 through about 26. So it will drop just ahead of menstruation. So it's going to be in the luteal phase. So that's going to be where it's really important. What does it do? For SL, you primarily just need to know its role in the menstrual cycle. HL, you need to know one other thing. So in terms of the menstrual cycle, its job is going to be to maintain the uterine lining. So we want to maintain the lining in hopes that implantation can occur. So it's going to be in charge of that. It is also going to negatively feedback FSH and LH. So it's essentially going to shut down the rest of the cycle so that no other follicles are developing until after that one has had a chance to implant for pregnancy. And then in the actual pregnancy stage, in pregnancy, it's also going to inhibit contractions. So it plays a role in preventing preterm labor by making sure that essentially while the placenta is producing lots of progesterone, it should prevent the uterine contractions that would lead to labor. So that's going to be its other important role there. Last one, and we will kind of mention it again when we do our next part, but the other hormone that you need to know at an HL level only is going to be oxytocin. Okay, so oxytocin is going to be released by it's actually released by the posterior pituitary, but is messaged because of cervical contractions. So this one's back to the pituitary. So this is our posterior pituitary. When is it released? So it is going to be released when the baby pushes on the cervix. And that's going to happen due to contractions. So if contractions happen, it pushes the baby's head on the cervix. That triggers the release of oxytocin. Oxytocin, what is it? Oh no, that was in blue and that should have been pink. Oh, my own color scheme. Sorry. What is it going to do? It is going to cause more contractions, which means it's going to release more oxytocin. So we can make a note that this is a positive feedback loop. So they've asked in the past, it's hard to know with the new course, quite a lot about the hormones involved. So I think if you can be really clear on like what it releases, when it releases, and then the other one is just to be aware of these feedback parts, because that can be really important. So what we have here is kind of a positive feedback, whereby estrogen produced by the growing follicles that were made by FSH release more FSH. So that's a real positive loop in the follicular phase. Then we have a negative feedback or inhibition pathway, whereby progesterone blocks that, and it also is inhibiting kind of contractions and therefore oxytocin. So positive feedback by estrogen, negative feedback by progesterone, and then of course the positive feedback loop of oxytocin. So know the hormones, who releases them when, but also be able to kind of account for which ones are a positive feedback mechanism, which ones are a negative feedback mechanism. Okay, so I have it next to this, but I'm going to erase just because I don't want to have to write in this super tiny space really small, but hopefully you have a bit of space next to it. It doesn't have to be super giant, to be honest. It can be a fairly small diagram. I'm just making some space for it. I'll leave these actually and just make a little bit more space here so that I can not have to write too tiny. Okay, so what I want going to do here is just kind of look at what's going on in each stage in terms of what's happening in the ovary, what's happening to the uterine lining. So I'm actually going to start at day 3. So day 3 is really the start of the follicular phase. The reason for that is day 1 is actually the day the period begins. So day kind of 26 to 3 is really kind of the menstrual part. So when the progesterone drops. So we're going to essentially start at day 3. And this stage from day 3 to about day 14. I'm going to do kind of day 12 to day 14 here. This is all going to be the follicular phase. So this is going to be follicular. And so again, we should already know, what does that mean? It means high FSH, high LH. So particularly LH is going to come here. So this follicular phase, and I didn't put this on mine, I'm just doing it now to really re-emphasize this, is going to be the stage that there's higher FSH, and then there's going to be higher estrogen as well. So both of those are rising. As a result, let's kind of show what's going on 2 places. So we're going to draw a little ovary, and then we're going to draw a little uterus. Okay, so those are our 2 things. In the ovary, what we would be seeing during this phase is the kind of growth of the follicle. So if we were to say, here's my little egg, and here's my little follicle. You may have to zoom in on your screens for this. By the end of this phase, it should be like a nice big follicle with a fully developed egg. So this is basically the development of the follicle. So the follicle is getting nice and big, the egg inside of it is maturing. So that's what's happening inside the ovary, is this development of the follicle. What's happening inside the uterus is it's getting a thicker lining. So it shouldn't be super thick yet, but it's getting there. So we're developing some lining in the uterus. Okay, so then day 12 to 14, our next stage, really is going to be here at day 14, but this stage here, kind of in between, is where my LH is surging. So the surge in LH at about day 12 leads to day 14, which is going to be ovulation. Okay, so what's happening now in our 2 places? So in terms of our egg, or ovary, sorry, our ovary is going to be releasing the egg. So this is going to be the stage of ovulation, where it's going to dump its nice mature ovum, or secondary oocyte, let's be honest. So it's going to drop its secondary oocyte into the fallopian tube. So release of the egg at ovulation. By this stage, because of the higher estrogen that's been building up, the lining should be even thicker. So nice thick lining. It's going to get even thicker still, but we'll give it a chance. All right, so nice thick lining in the uterus. Okay, so now from day 14 to about day 26, to day 28. So this whole stage here is going to be the luteal phase, all the way to day 28, but there's going to be that kind of 2-day window where the progesterone drops ahead of actual menstruation beginning. So that's why I'm just separating that. Same with day 12 to day 14. There's just a couple 2-day windows that are really important in terms of the kind of triggering. So during this whole phase, this is going to be the luteal phase. And this is all going to be marked by high progesterone. Like I said, the estrogen will continue during this phase as well. The other 2 will be completely inhibited. So progesterone is the boss of this stage. All right, let's look at what's going on in this stage in my 2 areas. So in my ovary, my egg has been released, and what I'm going to have left is the corpus luteum. So my corpus luteum is going to be left over. So that's basically the leftover follicle cells. So we see here, after they release the egg, they're going to become the corpus luteum, and that is going to be what's releasing progesterone. So this stage is all regulated by the corpus luteum. What's happening in the uterus? This needs to be the uterus's thickest phase. So this is where we are waiting for implantation. Implantation is probably not going to happen until about here. So in that middle time, you can just draw it like crazy thick. Just draw so much uterine lining. So lots and lots of uterine lining. The uterine lining began already in the follicular phase, but progesterone will generally thicken it even a little bit more and keep it thick. So there's my nice thick uterine lining. What happens at day 26 is the drop of progesterone. So progesterone will stop being made. And the reason for that is because my little corpus luteum dies. So inside my ovary, the corpus luteum is no longer, so it is empty. So we can say corpus luteum is gone, it has imploded on itself. So unless pregnancy has occurred, there is no longer progesterone being released. So the death of the corpus luteum. As a result, nothing is going to keep the lining in place anymore. And so the lining is going to strip down. So now you can show all of the lining coming down. So the lining is leaving the uterus and coming out. So then this last phase that's really happening between about day 28 and day 3, which is really only about 3 days. The menstrual cycle, usually bleeding continues through the follicular phase at the beginning, but that's really the key. And so that is going to be menstruation until the next stage starts. Okay, so again, that was just 2 different ways of showing it to help you kind of wrap your head around all of that. Okay, we're gonna do fertilization, which SLs probably should know as well, so stay for a little bit longer. And then HLs, we need to quickly, sorry, this section's really long, wrap up the rest of the cycle. All right. So what I want to look at here is essentially everything that happens kind of in the process of baby making, so to speak. So we've done all the other things. So what I want to do for HLs is really show it as 4 steps. For SLs, you only need to really know the first one, and probably not in as much detail as I'm going to teach it, but it's probably worth just listening just to get the main idea. So essentially, after the egg has been released, if pregnancy is going to happen, the next step needs to be fertilization. So fertilization, then fertilization will lead to embryo development until it becomes a blastocyst. Then once the blastocyst is big enough, it will develop, of course, into the embryo. But in terms of what do you next need to know, that would be the placenta. Leave enough room, yeah. And then finally, birth. So obviously these are not all of the steps, but these are the steps you need to be able to know about. And again, these 3 are all HL. So HL, HL, HL. Okay, and again, fertilization, I'm gonna teach it at an HL level. SL, you don't probably need to know the terms like acrosome reaction and cortical reaction, but you do need to know that the sperm penetrates it, so you can learn it with a little bit less terminology and you should be fine. Okay, so let's look at what happens in natural fertilization first, and we'll make a little note about IVF. Okay, so here's gonna be my egg kind of membrane, and there's some binding sites. So my binding sites are gonna be on the actual, and I'm gonna do, just to show you, this is the end of my page, but I'm gonna do like one below, so kind of each of these should be about half the space. Okay, so there's my binding site. Inside my egg, I have some DNA. So there's the haploid nucleus. I also have some cortical granules. And this came up in kind of specialized cells as well for HL. So it's always a good sign. They want you to know it when they put it in multiple topics. All right, so there's my cortical granules. There's my actual haploid nucleus. There's the binding sites. Sitting outside of that is going to be the zona pellucida. So this is going to be a jelly layer. So I want to kind of draw it looking a little bit not packed. So that's going to be the zona pellucida. And at this stage, it is jelly-like. However, it is blocking the actual binding sites. So when my sperm comes in, it is going to have to break that down. There's its nucleus. The way it does that is with some enzymes that were in an acrosome head. So the acrosome head is going to release enzymes that are going to break it down. So those are acrosome enzymes. That was on the sperm ahead of its head. It had a little acrosome cap. It releases the enzymes out of its acrosome cap, and then it's going to be able to get its way through the zona pellucida, and it's going to be able to get in there, and it will deliver just its DNA. So for the SLs, that's probably about all you have to know, is that, because you don't really need to know about the polyspermy. Once this happens, though, we want to make sure no other sperm get in. So we say prevent polyspermy. So what happens now, so here's my same thing. So there would have been other binding sites as well. So my cortical granules are all going to basically spill out their little enzymes. So these are cortical enzymes. So when the actual nucleus enters, so here comes this nucleus, and it is going to come over here. They're actually not going to immediately merge. They're going to sit as two nuclei and actually go through mitosis and DNA replication independently and then merge. So there's my two nuclei that are sitting there. Remember, this part never entered, so kind of the head and the tail are still out here on the outside. They did not actually enter, only the nucleus did. And then what happens is this zona pellucida, when it reacts to these cortical enzymes, forms a hardened layer. And at this point, the acrosomes could no longer break it down. So it's a two-step process. It starts with the acrosome reaction, which is the acrosome enzymes breaking down the zona pellucida. And then the cortical enzymes or cortical reaction is going to then harden the zona pellucida so that nothing else can get in. Okay, so that's fertilization. I just want to quickly, we're going to do it like super quick, but you do have to know kind of IVF as well. Only two really steps that are important. The rest, hopefully, I think you could work out. But just to revise, the first thing you have to do is you have to stop the normal cycle. So you give the pill usually, so the oral contraceptive pill, and you give that to stop the natural cycle so that the hormones bottom out so that you can manipulate it. 2, you're then going to have super ovulation, and that's going to be done with FSH, generally. Then you're going to have to retrieve eggs and collect sperm. And then 4, you're then going to do fertilize in the lab. And then 5, you're going to have to transfer back into a uterus. Okay, so those are essentially just the high points of that. SLs, you are done. HLs, we just need a couple more things. Sorry, this part's really long. There's a lot in it. Okay, so let's move on to the blastocyst. So you need to know the general idea of what a blastocyst looks like. So we have this outer layer, which is the trophoblast. The trophoblast is going to become the placenta. So that'll lead us nice into the placenta. And then inside, we have the inner cell mass, which is going to become the actual embryo. Like so. So what's going to happen is this is going to implant on the uterine wall, or the endometrium. And once it implants, it is going to release HCG. And the purpose of the HCG is going to be to keep the corpus luteum alive. So that is then going to release HCG, human chorion gonadotropin, is what that stands for. So I don't think you need to know that one as fully in name, because really the only dot point on that is more that you know how it's used in pregnancy testing. But it's going to release that, and then that is going to keep the zona pellucida alive and tell the ovaries to keep producing the progesterone until the placenta is ready to take over. So that's going to be important. We're going to do pregnancy testing in a minute. We'll do it first, and then we'll do placenta. Okay, so it is going to release this hormone called HCG. HCG is only released during pregnancy, so it's a great way to test for pregnancy because that is the only time that it should be around. So it is used in pregnancy testing. Okay, and then let's look at how it would be used in pregnancy testing. This can go across to the placenta. We don't have that much we need to do on the placenta. So basically what is made are these monoclonal antibodies. So monoclonal antibodies that specifically bind to HCG. So my monoclonal antibodies are going to bind specifically to HCG. And they also have on them dye. So that's going to be made in the urine. Then up here, there's going to be anti-ones that are matching them. So these are going to try to also bind to HCG. So if that happens, and these are activated as well, it leads to a chemical reaction that releases the dye, and you get a line. So that is going to release the dye if both are there. So if the HCG is present, it attaches these with dye on them to the ones in the stationary spot, and that is going to cause the dye to be released and cause the color change. I've drawn it super clearly here. But essentially, when it hits both places, these are going to be reacting, and then my dye is going to be released. So that is going to be the line if HCG is present. Further down, you're going to have other antibodies, and they are actually not HCG specific. They are compatible just to the antibody itself. So in this case, it can bind like this, and it doesn't matter if there's HCG or not. Either way is going to trigger the color change. So in this case, it's going to happen whether HCG is present or not. So I'll go ahead and draw HCG on one of them just because I'm using the example of someone who is pregnant, so there would be HCG. But one could bind without HCG as well. And again, once this binds, that causes the reaction that releases the dye. So this is the control line and does not require HCG. It's going to happen if HCG is there or not. Okay, so again, I went through it pretty fast, but But basically knowing the idea that these special antibodies, called monoclonal antibodies, are made, and that the way it works is by essentially having the antibodies bind to HCG, and if they double bind to the ones that are kind of positioned in that spot, then you get the release, and that's how you can tell that someone is pregnant. So that is kind of the idea. These are kind of additional antibodies that are stationary. So the other ones are going to be in the actual moving through the urine, and then those are going to be the stationary ones. Okay, placenta, we're getting there. So with the placenta, just quickly, so we're going to have the fetal blood supply. Like so. And then what it does is it goes off into these little clusters that have kind of these projections to make them small. So these are called placental villi. So you do need to know that term. And those are going to be high in surface area. So high surface area, and that's going to enable fast diffusion. Meanwhile, over here, we have maternal blood that is also coming through. So this is the maternal blood. And interestingly, the maternal blood is the one like loose blood. So it's just going to come here and fill in all the space. So that's just going to be loose maternal blood that's all near the fetal blood, and that's going to allow for the diffusion. So you've got fetal blood, maternal blood. The fetal blood stays inside capillaries. The maternal blood actually exits vessels to allow for the diffusion, and then will be collected back after it's kind of given up a lot of its oxygen and returned. That's all you really have to know about the placenta. It's just the villi for surface area, and that is where diffusion of materials is. All right, last thing is birth. So let's look at what happens for birth. So we are going to have a drop in progesterone. That's going to happen as the placenta ages. So when placentas get older and reach that kind of 40 weeks, they will stop releasing as much progesterone. Plus, you're also going to have a big baby. So plus a large baby. So when you have both of those factors, the result is going to be that there's going to start to be some contractions. So you don't have the progesterone blocking it, and the baby's getting really big. Those contractions are going to push on the cervix. Pushing on the cervix is then going to trigger the release of oxytocin. Releasing oxytocin is going to cause more contractions. More contractions is going to cause more oxytocin. So again, we just want to remember that this is a positive feedback loop. So it's going to keep going until the baby is born, in which case there is no baby to push on the cervix anymore, and the process is done. So that's essentially the walking you through that. Okay, so it's really long. I think the other two sections will be shorter, but that's everything you need on reproduction. Okay, so we're going to do D3.3 now and look at homeostasis. So this is going to be a pretty all SL, all HL, so it's a little bit more removed. So the SL stuff is going to be the thermoregulation and glucose regulation, and then the HL stuff will be what's going on in the kidneys. So we'll basically do half a page of each. So try to keep your SL stuff to half a page HL, so you have the bottom for your giant nephron. And SLs, you just have less to cover, so all good. So before we go into the nitty gritty, I just want to make sure this should be obvious, but just do a couple of really important definitions. So I want to make sure, A, that we know the definition of homeostasis, and B, that we can connect it to negative feedback loops, because that is one of the dot points. So if we start with just defining it, and it's kind of a strange time to define it, because we've been talking about it previously in other contexts. But if you look at C3, the emphasis on endocrine system was more on hormones that are regulating other kind of processes in terms of growth and fight or flight, things like that, melatonin, whereas this is really now zooming in and looking specifically at what we're looking at in terms of hormones that are involved in homeostasis. So what is homeostasis? It's the maintenance of a constant internal environment. So it's maintaining a consistent internal environment despite changes in the external environment. So all organisms strive to do this in some ways, but our emphasis will definitely be on kind of for humans. There are 4 different things that are kept in kind of pretty tight homeostatic balance. So 4 things that are particularly important, and you will have to know all 4, at least for HL, SL a little bit less on the osmolarity. So the examples of the things that you need to be able to explain that for is body temperature. So we're going to do that now. Blood pH, which you should already know because we did that in C3. So looking at that idea of the role of the medulla and chemoreceptors and adjusting heart rate and ventilation rate in response to that. So you need to know that. Blood glucose, which we're going to do now. And then osmolarity of the blood. So osmolarity for HLs, you have to know the specifics. For SLs, you just need to know kind of what we did in C3 as an example of something that the hypothalamus and the pituitary regulate, and know ADH is kind of a general term for the hormone that is involved in that. Okay, so, that was the other thing I was going to add. So that is what homeostasis is. The other thing that the course really wants you to be aware of is that when we regulate homeostasis, we do that with negative feedback loops. So the maintenance of an internal environment, and that's done by using negative feedback loops. So positive feedback loops are generally not involved so much in homeostasis. They have roles in other processes. But when we think homeostasis, we're definitely thinking negative feedback. And the idea of a negative feedback loop, the way they work is essentially they make small adjustments to return to a set point. So the way negative feedback loops function is around this idea of returning to a set point and then stopping the cycle. So return, stop, return, stop. So that's how negative feedback loops work. And both glucose regulation and the thermoregulation are both examples of negative feedback loops. So if you were asked about describing a negative feedback loop, these would be great choices to do. You could also do it for osmolarity and ADH. You could also do it for blood pH in terms of adjusting respiratory rate. So all of those work under negative feedback loops. Okay, so we're gonna do blood glucose first. So I'm gonna do it similar to how I did the sex hormones as a table, just to kind of, rather than do it as a giant diagram, really emphasize kind of the 2 hormones and their role. So our next little table is gonna say hormonal control of blood glucose levels. And then we're going to leave a little bit of room. So we'll make our table and then a little room to the side. There's not many disorders they specify you really need to know about, but they have specified diabetes as something you do need to know about. So we will leave a bit of space over here to talk about the disorder when that is not done easily in the body. So you know how it should work and then what the consequences are if it doesn't work like it should. So just 2 hormones, a short little table. So we're going to say, what is the hormone? So it's actually going to be very similar to our sex hormone table. So anytime you're learning hormones, you should be able to think who releases it, when is it released, what does it do? So you need to be able to answer those 3 things. So hormone released by, you need to be able to know when is it released, and then you need to be able to know what are its actions. So again, essentially any hormone, when you're doing your final revising, those are good things to kind of check. can I account for that for all of them? Okay, so first hormone we'll look at is insulin. So insulin. Insulin and glucagon are both released from the pancreas. So a new organ, we've done lots of pituitary. So this is in a different endocrine organ. And so the pancreas also makes some digestive enzymes, which you don't have to know about, but it's not purely an endocrine gland. So it has clusters of cells inside the pancreas that just work on releasing these hormones. So it's going to be released by what are called beta cells. And they're in these specialized areas of the pancreas called the islets of Langerhans. And you need to know that that is in the pancreas. The specific thing to remember that is unique between the two is the beta cells. But if you just say beta cells and have no idea where they are, you're not going to get all your points. So knowing that information. We release insulin when blood glucose levels are high. So after meals. And the action of insulin is going to be to reduce blood sugar levels. And it's a little bit more complicated than glucagon. So you need to know that it's done at least two of the ways. I'm going to add the third way because I think it's worth bearing that in mind as well, but definitely the first two. So the first and most important action of insulin is to make sure your cells take up that glucose so they can use it for cell respiration. So they're going to trigger cellular uptake. So they're going to tell your body cells, please take up some of this glucose and use it, please. So that's going to be their first kind of idea of how to reduce it, is to get it to where it needs to go. Once your cells have taken in the glucose they need, the next thing that you're going to do is try to store it. So then you're going to store the glucose, and you're going to store in the liver as glycogen. So it's going to get converted from glucose to glycogen in the liver. The third option is also a storage, and that's just going to be to convert to fat. So essentially kind of the three things work together to overall lower it. The course doesn't specifically say about converting to fat, so you could just cover, definitely need to know the cellular uptake and the liver as your kind of primary ones. Okay, so those are the responses to blood glucose levels being high. We have a different hormone that responds to blood glucose levels being low, which is glucagon. So glucagon also released by the islets of Langerhans in the pancreas, but it's going to be released by alpha cells. So alpha cells in the islets of Langerhans. And I'm just shortening that because we know it's in the same area. So different set of cells. This time, this is going to be released when blood glucose levels are low. And so it is going to have the opposite effect. So it is going to raise your blood glucose levels. And it's going to do that in one primary way. So it is going to act specifically on the liver. And the way it does that is it tells the liver to convert glycogen, which we just made in the step above, back into glucose. And remember, glycogen is a short-term glucose storage. So it is not intended to store glucose for long periods of time. It is really that kind of overnight store. So it basically stores glucose so you could use it the next day. It's not long-term. Fat is for long-term storage. Okay, so if you have a disorder related to your inability to regulate glucose, it could be two things. Hypoglycemia is actually that actually relates to hypoglycemia relates to low blood sugar. So that's a different kind of set of disorder. The one that you need to know about is diabetes. So diabetes is a disorder related to blood sugar regulation. We talked about the fact that regardless of whether you have type 1 or type 2, the symptoms are actually very similar. So the symptoms are going to be things like fatigue, extreme thirst, frequent urination, blurry vision, things like that. I always think of it as two separate set of symptoms. The first one is related to low sugar inside your cells. So that's going to cause like the fatigue and weight loss. So your cells are not utilizing sugar. So that's causing you to have low energy. The other set of symptoms are related to the glucose staying in your blood, which is causing your blood to become hypertonic. So that's causing this kind of high sensation for thirst, the kidney constantly trying to kind of process. All of those symptoms are related to that. So that's kind of your two set of symptoms. You do have to know about the two types. So type 1 versus type 2. So type 1 is an actual autoimmune disease. So it's autoimmune destruction of the beta cells. So it actually kills the beta cells, and this generally happens in childhood. So it's quite rapid onset, whereby as long as you have some, they're still functioning, but generally the immune system just something flicks and it starts attacking them. So the consequence of the destruction of the beta cells is there is no insulin produced. And generally speaking, these individuals would be insulin dependent, so they would need a pump, something to create the insulin for them. So that's type 1. Type 2 diabetes is basically a desensitization to insulin. So it generally happens when the cells stop responding to insulin. So there's still insulin present. It's just that the cells stop listening to it. So it actually can be a little bit trickier to treat because you can't just give insulin. So you have to work on kind of receptors and so forth. And this happens during adulthood, often due to kind of a number of risk factors, but kind of long-term chronic exposure to sugar can lead to it. So it can be linked to kind of diet and things like that, but it also can be linked to other health problems. It can also just be linked to genetics, things like that. Okay, so that's really all you need to know is the difference between those two in terms of what causes it and when the onset is. Okay, so I'm gonna move straight from that. I'm gonna erase. So next little section here, we're gonna look at thermoregulation. All right. Okay, so thermoregulation is something that's really only done by endotherms. So ectotherms do not worry so much about having their internal temperature be constant. So it's an endotherm reaction, but the actual ones that we're going to talk about are the human responses. So we're going to put thermoregulation and endotherms, but we are not going to discuss how birds react. We're going to focus in then on humans. So the concept belongs to endotherms, but I want to make the point that we are going to specifically look at kind of human responses. So for instance, we're actually not going to put like goosebumps because that's usually more of a mammalian response that has hair stand up to kind of trap the cold out. But because we don't have body fur, it actually is not effective for us. So we're going to leave that off. All right, so we're going to cover 6 total responses, 2 to heat and 4 to cold. So use your space unevenly. So 2 responses to heat and 4 responses to cold. So to heat or to rise in body temperature. This can happen for other reasons, including like high metabolic rate. So interestingly, the cold is basically solely due to the environment, whereas heat can happen because our own metabolic rate is high. It can happen because of fever, so kind of different reasons for that. So only 2 that you have to know for heat, and they should be pretty straightforward. So the first one is to create sweat. And the key with sweat is it actually releases heat when it evaporates. So heat released at evaporation So essentially it uses energy or heat to get it to evaporate, and that releases it from the body. That links back to what we talked a little bit about properties of water in terms of high heat of vaporization. So the process of getting it to vaporize pulls heat for the energy for that process. So that's kind of why it works. So again, just kind of clarifying that really the cooling mechanism happens at the evaporation stage. That's actually the release. It's not just that you feel cooler because you're wet. Second one, and we have to know this for both, which way goes which. So this time we want to move the blood close to the surface. So if you're hot, you often have pink cheeks. You look warm. So your blood's going to go to the surface so that it can then basically leave out to the external environment. So in order to make that happen, we're going to have vasodilation. So vasodilation means the arteries get nice and big, allowing the blood to go to the surface capillaries. So the vasodilation is of the arteries. That is going to send blood to the surface capillaries, which is why you're going to appear red. And then when you send blood to the surface capillaries, heat automatically transfers from one environment to the next. So this is going to allow the heat to come off of your hot body temperature out. This only works if the atmospheric temperature is lower than your body temperature. So this isn't going to work great on a sweltering 40-degree day, but it's very effective even on a 35-degree day if you've overheated. So then it is going to be heat lost from the surface to the environment. So the heat, once you pull the blood close, then that actually enables more of the heat that's in your blood to come out to the environment. Okay, so that's vasodilation. So those are your two responses. Then we're going to say, what are our responses to cold or to a drop in body temperature? And this is generally due to environment. So we do not have a lot of endothermic reactions in our body. So our metabolism doesn't generally drop our body temperature, but cold environments do. And most humans live in environments that generally fall under 37 degrees. So other than some very hot days, most of the time our external environment is colder than our internal. So this is kind of a constant thing that we have to manage. Okay, so 4 different responses here. We'll come with the one that comes straight off of this. So number 1 is going to be vasoconstriction. So vasoconstriction, this time we don't want to lose heat. So our body temperature is warmer than the environment. So if our blood is close to the surface, the heat will go out. So we're going to constrict the arteries. That's going to keep blood away from the skin. And that's going to hold on to our heat. So we want to keep the blood out of our surface capillaries, and that is going to reduce heat loss. So that's not necessarily going to make us warmer, but it is going to hold on to the heat that we have. It's also why you're going to look pale or even a bit blue if you are cold, so because you pulled all your blood in to protect it. Okay, next one, and this is going to kind of relate to the one after it, is that we actually want to accelerate our metabolism. So this just keeps your heat. We want to make some heat. So in order to make some heat, we need to increase metabolism. So I'm just going to write as the reminder thyroxine. So thyroxine is going to do that. You do need to kind of know the steps involved in this. So first of all, your thermoreceptors are going to send messages to your hypothalamus. So your external skin receptors are going to say, help, I'm cold. And then that's going to go to your hypothalamus. So thermoreceptors to the hypothalamus. Your hypothalamus is then going to send a message to its close personal friend, the pituitary. That'll be your anterior pituitary. Your anterior pituitary is then going to release thyroid releasing hormone, or thyroid stimulating hormone, sorry. So TSH. That is then going to travel to your thyroid gland. Your thyroid gland is then going to release thyroxine, which is a hormone. And that is going to cause an increase in metabolic rate. So that is going to tell your body to just increase your metabolism, because when we are doing metabolic processes, even normal cellular respiration, heat is often a byproduct. So it's going to increase your resting metabolic rate, so even when you're sitting still. Next one is related to that. So in order to increase your metabolic rate, it can increase your just baseline, but also it's going to try to encourage muscle use, because muscle use is going to actually create more cellular respiration to fire the muscles, which creates more heat. So it's going to cause your muscles to become active, which can, hopefully we can behaviorally think, oh, it'd be a good idea to go for a run. But if you don't work that out, your body works that out for you by making you shiver. So number 3 is going to be to shiver. So shivering is actual muscle contractions. So these are going to be muscle contractions. If your muscles are contracting, that is going to increase your demand for energy, and so that is going to increase your rate of metabolism. So you're going to have to essentially do more cellular respiration in order to maintain those little contractions, which is going to help you to burn up more of your food, which releases heat as a waste product during cellular respiration. So you get heat that way. So this is, again, both of these things together are going to actually create heat. So they are going to actually create or release heat into the body when that happens. The third one is actually probably more specific to babies, but we have an ability to do it in some respects. So the fourth thing that we're going to do, so I've said previously that even normal metabolism will release heat as a waste product. So cellular respiration is not perfect, and some of the actual transport of ions or hydrogen protons will actually create heat rather than ATP. But normally the process is biased towards trying to create ATP and not heat. So we have special tissue that does a specific form of cellular respiration that makes heat and not ATP. And that process, that you need to know the name of it, is called uncoupled cellular respiration. We'll go into a little bit more when we get to cellular respiration, but this is only done in brown adipose tissue. So this is a certain type of fat tissue that actually creates heat. So in humans, we see that primarily in babies. We have some of it left as an adult, but not a lot, because we tend to have other mechanisms that we can use. But for babies who really don't shiver or not as active and have really small surface or high surface area to volume ratios, they need a little bit more help. So they have brown adipose cells. And in this case, ETC, which is electron transport chain, creates heat rather than ATP. So babies have lots of it. We lose it kind of throughout our lifetime, but it's a good mechanism for those who are a little bit more vulnerable to the cold. Okay, so that's everything SL, so you guys are good to go. HL, we need to pick up on the kidney. Race, race, race. So I did a lot of tables there. So what I'm going to actually do, just to try to show you, is we're actually going to draw one giant nephron across and kind of label a few things in the sections, and then below it try to say what's happening. So leave us some space below for your descriptions, but draw a nice big nephron. So if you draw a tiny nephron, you're going to have awkward space left at the end. So just to kind of clarify that, I will try to make my board represent the same ratios. Okay, so they want you to specify, which I don't know that we talked a lot about in class, so we did it in like the homework, but I'll kind of hit it here, is that when we talk about the kidney, there's actually two things going on. So one is the osmoregulation piece, which is really what goes with this homeostasis unit. The other is the excretion, which is just getting rid of waste. So both of those are happening in the kidney. So we're just going to put that as our title, is osmoregulation and excretion in the kidney. Kidneys. I'm bad about forgetting other people have two. So this is HL. HO. Great, good on that. And this is going to happen at a single nephron. So at each nephron. So a single nephron does the whole process, and then that happens like a million times. So it's just like filter, 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 filter. So there's a whole line of filters. So we're going to say at each nephron. The emphasis is really on the nephron. The old course had a bit more on kidneys. They don't really have kidneys in the new course. It's really just the zoomed in. So I'm just going to draw a tiny kidney up here just to know the two terms I want you to know is the medulla versus the cortex. That's the only thing that kind of comes up. It's not a very pretty kidney, sorry. So this inner part, which is where all your loop of Henleys are, that's called the medulla. So that's like the inside of the kidney. The top part, which is where all of your glomeruli are, is the cortex. So it's probably the only two parts you need to understand is that there is the two layers. So the top is the filters, the bottom is the loop of Henle and collecting duct. So we kind of do the actual excretion here, the osmoregulation here. Okay, so like I said, I'm going to draw the whole nephron across first, just so we can kind of space it. Then we'll go back and do some labeling and clarifying some things. Picking my colors. Thank you. All right, so let's start with the glomerulus. So I'm going to start here. Okay. So I want to try to draw my afferent arteriole looking bigger than my efferent. Then I'm going to draw some kind of cluster of capillaries through here. And then I want to put some fenestrations in them. So try to leave out some fenestrations to show that there's little holes in this. Okay? Around that, I need another marker, I forgot. Around that, let's draw a basement membrane. And it doesn't look like the new course is very concerned about podocytes, but for my class, I have a grievance with the podocytes. So I'm going to draw some podocytes, because if you don't have them, bad things happen. There you go. So there's your podocytes. Around the glomerulus, so that whole thing is the glomerulus, and I probably should have started a little bit lower, but it's all right. This is the glomerulus. And then around it is Bowman's capsule. So Bowman's capsule surrounds the glomerulus, like so. So it's going to be around the whole glomerulus. So that's going to be Bowman's capsule. Okay? From Bowman's capsule, it's then going to go into the proximal convoluted tubule. Oh, really? Proximal convoluted tubule. Okay? From the proximal convoluted tubule, it is then going to go down into the loop of Henle. Should be a little bit more upright. So I still want some room to write about it below it, so don't go all the way down. This is not really quite to scale because it should actually be lower than the glomerulus. So it would actually go quite a bit lower, but I just drew a really giant glomerulus, so these things happen. So there's my loop of Henle. So that's my next stage is the loop of Henle. From there, it's going to go to the distal convoluted tubule, which you don't have to know anything about, which is kind of them. I'm just abbreviating distal convoluted tubule. And then the last step is that it then goes from there into the collecting duct, which you do have to know about. All right, so those are the parts of the nephron. So we'll add a few more things into the details of it as we go. And then we'll kind of, like I said, right underneath what's happening at each stage. Okay, so let's start with the glomerulus. So the glomerulus is really going to be where excretion happens. So this is essentially excretion. This is all osmoregulation. So we'll kind of clarify that. So first of all, the idea here is going to be, this is going to be the site of ultrafiltration. So we're going to get all the small things out. For that to happen, the afferent arteriole that's bringing it in has to be thicker than the efferent arteriole that is collecting it after. So the small efferent compared to the afferent is going to increase causes pressure. That pressure is going to be what causes ultrafiltration. Ultrafiltration is when the things squeeze out. And in order for them to squeeze out, they have to go through these little fenestrations, which are going to be the little pores that allow them to get out. And then we want to make sure that only small things get out because there is not reabsorption for proteins. So then we're going to have here the basement membrane, which is going to help with trapping the proteins, as well as the podocytes. So those together are going to be in charge of making sure actual blood cells, but also proteins do not get out. Okay, so all of this together, the main purpose of this section is ultrafiltration. So ultrafiltration is done by size. And the purpose of ultrafiltration is the excretion of waste. So this is where excretion or removal of waste occurs. So when we say by size, essentially the small molecules leave the glomeruli into Bowman's capsule. So small molecules are squeezed into Bowman's capsule. So Bowman's capsule is the orange in my diagram, kind of area underneath it. Once it is in Bowman's capsule, that collection of small molecules is now called filtrate. It has a number of different things in it. So if we look at what would be in filtrate, it would be anything that's small, which would be water. It would actually be waste products like urea. It's going to have glucose in it. It's going to have salts and stuff in it, all of those things. So those are all small, and that is all going to make up filtrate. Some of those are good things that we're going to have to reabsorb. What should not be in there is proteins. So by size, so we can make a little note, but not proteins. Proteins are big, so they should stay in. So that's the purpose of the basement membrane, is to keep the proteins in. Okay, so that is ultrafiltration. Because we removed some good things, so these are good things, I want them back. We are now going to have our next phase. I had more space in my diagram. I've not done a great job here. We are now going to have a selective reabsorption. And that is going to happen at the proximal convoluted tubule. I'm going to draw just above here, just a reminder that these cells, like inside here, that would be an example of a cell, are going to have the microvilli. And that's just going to increase the surface area. They're also going to have lots of mitochondria. Okay, so this we are now going to basically remove. So we're going to pull the small molecules. So small good molecules are returned to the blood. There's a lot of small good molecules that are returned to the blood here. The primary one that you need to know about, well, about 70% of the water is actually returned here. So it's a really big place for return of water, but also for glucose. So that 100% of the glucose needs to be reabsorbed here. So that's a really key one. And remember, that is done with sodium glucose co-transport. So Na glucose co-transport. That's actually in A2. B2 with membranes. So that's actually not in this topic, but it's relevant, so revisit that. But basically the idea of moving sodium by active transport to allow glucose to then be moved with the concentration gradient when the sodium comes back in. So I'm not going to go into that because this video is already going to be super long, but do revise that idea that it's basically active transport of sodium to allow for the passive transport of glucose. And that's what's happening here at the proximal convoluted tubule. The other thing I wouldn't mind if you want to just draw some little blood. Woo! There is kind of blood all floating throughout this so that things can get back in. These are called peritubule capillaries, which is basically the blood flow that's running all along it. There'd probably be more of those, but just to clarify that when we're reabsorbing it, there's kind of blood flow all around it. Okay, so now onto the loop of Henle. I covered this in more detail in class than you need, so I'm going to try to cover it in less detail here with really focusing on the overall kind of point. So we do have the two limbs. So here's my filtrate. So this is now filtrate. So it's come down here, through here, and it's going to go down and then ultimately up. So that's where these names come from. So this is the descending limb. I'll move that. That just refers to the fact that it's going down. And then this is the ascending limb. That just refers to the fact that the filtrate is going up. Okay, so descending and ascending limbs. So what's happening here? So essentially two things are being removed from the filtrate and they're happening on opposite sides. So on this side, we are going to be removing water. So H2O. So water is being reabsorbed. So what is the main purpose of the loop of Henle is water reabsorption. So we're going to reabsorb the water, the remaining water. The only way that's going to happen, so that's going to be reabsorbed by osmosis. So that's going to be a passive transport process that's going to go with the concentration gradient. So removal of water reabsorption by osmosis from the descending limb. But the only way that's going to be possible is if the water potential or the hypertonicity is higher in the medulla. So what needs to happen for this to be the case is that every point along here needs to have the higher water potential. So every point in the filtrate needs to have higher water potential. The way that is managed is by active transport of salt from this end. So what we're going to do is we're going to pump out salt into this area, into the medulla, and so that is going to keep the medulla hypertonic, which is going to pull out the water. This process is active, particularly up high. It's a little bit confusing. Essentially at the lower point, the actual salt concentration is so high in the filtrate, it will go out. But as it gets higher up, it actually has to use active transport to pull the salt out. So active transport there to facilitate this. So osmosis of water from the descending limb, but the only way that is going to be possible is due to a hypertonic medulla. So this is the medulla. So this whole area all around it is the medulla. So that's just the tissue surrounding the loop of Henle. So that means the medulla, which has all the blood running through it. So the medulla and the capillaries need to be hypertonic. So that is going to be due to a hypertonic or low water potential. Use whatever wording works for you. Medulla. And the way that we ensure that medulla is in fact hypertonic is maintained by the active transport of salt out of the ascending limb. Okay, so these two things are working together. So this is making it salty. This allows the water to leave. So you need both of those two processes to happen to keep that managed. And again, this is primarily kind of a standard thing that's happening no matter what. The last step then is really where the osmoregulation happens. So the last step is where we're actually going to see water reabsorb depending on the person's individual needs. So this is going to be done by ADH. So over here, we are going to have ADH receptors. So inside the cells themselves, and this is a slight oversimplification because it's really only on like the walls, like that, if you can imagine. But I'm just going to kind of show the general idea. So what we are going to end up having is we are going to have these little vesicles, and inside the vesicles are going to be aquaporins. So that's going to be a little vesicle of aquaporins. So if you are well hydrated, then your water has already been returned because of the loop of Henle. So if you're well hydrated, any remaining water in your urine at this point, you can just pee out. So you don't need it, you're well hydrated. But if you're dehydrated, your body is gonna send a message in the form of ADH. So ADH is gonna come over here and it's gonna bind to an ADH receptor. So that is essentially what those are, ADH receptors. The ADH is actually coming from the pituitary, from the posterior pituitary. It's the anterior normally gets all the glory. So from the posterior pituitary, and it's being released in response to high solute concentrations. So that's going to be when osmoreceptors in the hypothalamus detect high solutes. Okay, so that's our little bit, we already have talked about that, so that shouldn't be brand new information. So posterior pituitary, osmoreceptors, hypothalamus, high solutes. When that happens, it causes these vesicles to come and merge to the side, and it's going to cause the placement of aquaporins in the cell membranes. So this vesicle is going to come and merge with the actual membrane when ADH is present. So only when ADH is present. So when ADH is present, it's going to say, hey, come merge that. If that has been merged, then what we end up with is aquaporins along the wall. And what that means is, as the filtrate is moving through, so filtrate is continuing to move through, the water from it is going to leave. So we're going to end up reabsorbing more water out of the filtrate and back into ultimately the blood supply. So water is going to leave the filtrate if that happens. So this process is basically maintaining homeostasis, and it does that by adjusting water reabsorption based on hydration status. So depending on how hydrated you are, that is going to alter that. Now, once this is done, it is now no longer filtrate, it is now urine. So at the end of this process, in theory, it is now urine, and it will go and connect to the ureter. And then the blood that's left here will go through the next one. So kind of at the end of each nephron, we have made some urine, and then it will go through the next bodily blood, will pull more things and make more urine. Okay, so I think that's the main idea, is that you can kind of account for what's going on at each step. So glomerulus, ultrafiltration, proximal convoluted tubule, selective reabsorption, loop of Henle, water reabsorption, collecting duct, osmoregulation. So being able to say what's happening at each stage. All right, thanks.